So it's a pleasure to introduce today's speaker, Dr. Andrew Nash. Andrew is currently Senior Vice President for Research at CSL and is based at Bio21 Institute, just around the corner. He's told me to keep it simple. He's going to talk to us about development of new therapeutics and going from discovery through to the clinic. And we're very interested to hear what he's got to say. And it is short.
Thanks, Sandra. So as Sandra said today, I'm going to try and give you all some insight into what it takes, the process involved in moving an early stage discovery project from the lab bench where you all work through to the clinic. And I guess with the projects I'm going to look at, it's not just through to the early stages of clinical studies, but through to phase 2 and phase 3 studies as well. So in a sense, that process of moving a project from the bench to the clinic is a bit context dependent. It's different for different companies. It's different for small biotechs compared to mid-sized pharma compared to large pharmaceutical companies. I'll be giving you a CSL perspective. So in that sense, I will spend at least a couple of slides giving you an introduction to CSL and the process that we go through in order to decide that a compound or a molecule warrants progression through into clinical studies. And I'm going to then use, I guess, 3 different projects to give you 3 different insights into how that process travels along. So firstly, I'm going to talk about a compound that we refer to as CSL 654. It's a recombinant coagulation factor in the clinic for the treatment of Haemophilia B, and it's in phase 3 studies now. I'm then going to talk about CSL 312. It's a monoclonal antibody targeting factor 12, and we think it's going to be very useful targeting some rare genetic deficiencies and in the bleeding thrombosis space. It's just been through the preclinical workup, lead development preclinical workup phase, and is just now moving through into toxicology and clinical studies. And it's a great example of the level of data these days that you have to generate to justify making that big step of moving a compound through into the clinic. And finally, I'm going to touch on CSL 362, which again is another therapeutic antibody. It's currently in the clinic in phase 1 studies for oncology and autoimmune disease. But I think this is a really good example of where if you persist, hopefully you can have success in the end. So this molecule actually has started on the bench, it's been into the clinic, it's failed into the clinic, it's been back to the bench for some re-engineering, and it's gone back to the clinic again now. So it's a great example of what you can achieve if you persist and gain a better understanding of the disease you're trying to target and the mechanisms that are going to be required to achieve that. So firstly, just in terms of some background, so I will tell you a little bit about CSL. CSL is a global company now. It always surprises me the limited extent to which CSL is understood, particularly within the biomedical research community in Australia and even in Melbourne. It's Australia's 10th largest company. Globally, we employ about 11,000 people. Our market capitalisation is in excess of $30 billion at the moment, and we have annual sales of around $5 billion. And you can see the countries in which we operate here. So we're still, we're still an Australian company, and our headquarters are here in Melbourne, just up in Poplar Road behind the zoo there. But now most of our activity, most of our sales and most of our workforce is in the Northern Hemisphere. And we do have large-scale manufacturing facilities in Switzerland, in Bern, in Germany, in Marburg, and in the US in Kankakee, which is just south of Chicago. And in fact, most of our Northern Hemisphere operations are run out of a big head office in King of Prussia, just outside Philadelphia. So that's what CSL looks like now. It's a big change from when the company first started in 1916, and even when the company first listed on the stock exchange back in the early 90s, when we had a couple of thousand employees and a market cap of about $200 million. So the company has certainly grown extraordinarily over the last 10 to 15 years. And that's a great thing, because what that has allowed us to do is to spend a lot more money on research and development. And that's what I'm interested in, and I suspect that's what all of you are interested in as well. When I first started at CSL 7 years ago now, back in the end of 2006, the R&D budget was about $140 million. And that sounds like a lot, but it doesn't really get you that far. Since that time, in the last 7 years, that R&D budget has grown from around that 140 million through to this year we'll spend close to $500 million on R&D. So that's a really rapid growth, and it reflects the growth in the company over that period of time. So a fantastic outcome for those of us that are working in R&D. And you can see here on this slide that we really spend our R&D dollars in three different areas. And this is important when you're considering taking new molecules through into the clinic. So we spend money on life cycle management. That's taking our current products and looking at other indications, new areas, new ways in which we can use those current products to treat diseases. We do market development, which is again, pushing our products out into new markets and looking to do new things with them, generate further revenue. But you can see that the growing piece of this puzzle and the one where I work is in new product development. So over the last, probably 5 to 10 years, our level of investment in lifecycle management, market development has really stayed steady, while our investment in new product development has grown a lot. And it's that new product development which we're talking about today. It's about taking new molecules from the bench and through into the clinic. In terms of the sorts of areas in which we work in CSL, it does to a large extent reflect where we're successful out in the marketplace, but it does include new areas as well. So CSL is very successful, and I will talk a little bit more about each of these areas in just a tick, but if you look at our product portfolio, immunoglobulins, haemophilia products, specialty products are all very important to us and form a large part of our R&D effort. And then we have this other catch-all called breakthrough medicines, where these are new projects which may fit within some of these categories or may not, but which are generally new mechanisms of action in new disease. So the R&D strategy is to maintain a commitment to extracting the maximum value from our existing assets and supporting the improvement of those products. But it's also to develop new protein-based therapies for the treatment of serious human diseases. So I think that's an important point for us as well. In terms of your ability to develop a drug through from the bench to the clinic and then beyond, you really do have to stick to what you know, to where you have the expertise. All of our expertise is in protein-based therapies, so our portfolio consists exclusively of protein-based medicines, really not small molecules at all. And if you think about the diseases in which we work, they're all serious diseases, and in general, they're all very rare diseases as well. So this is another thing that's specific to CSL. So just to touch on each of these areas, when I talk about immunoglobulins, I'm talking about the intravenous and subcutaneous delivery of plasma-derived polyclonal Ig. So all of those manufacturing facilities I talked about, they collect around 10 million litres of plasma a year, and they fractionate that plasma into a variety of products, and the most important product from that plasma is immunoglobulins. That's used to treat primary immune deficiencies, autoimmune disease and neurological disease. So we have quite a bit of research going on in that space. In terms of haemophilia, from that plasma you also extract plasma-derived coagulation factors, but we also are involved in the development of recombinant coagulation factors, and I will talk about one of those projects specifically. But they're for the treatment of things like haemophilia A, haemophilia B, and von Willebrand's disease. The specialty products, again, these are primarily plasma proteins, but are starting to include things like recombinant antibodies, and they're almost invariably for the treatment of rare congenital diseases. So, for example, people with deficiencies in things like C1 esterase inhibitor and alpha-1 antitrypsin. Also included in that list of products are really products to treat acquired bleeding disorders, and they're generally complex multi-component products. And when I talk about acquired bleeding disorders, I'm talking about people, for example, who are being treated with warfarin and need emergency surgery or surgery for any reason, and you have to be able to reverse the effect of that warfarin, so we generate products for that. In terms of the breakthrough medicines, and again, this is the area which is of great interest to us, It's again about plasma proteins and recombinant antibodies. It's generally now about novel therapies and new mechanisms of action. And the areas in which we work, although they're quite diverse, I guess you could describe them as falling into the oncology space, the autoimmune space and the cardiovascular disease. So that's the R&D strategy. And I guess the other important point to consider in thinking about projects moving from discovery through into the clinic is, for a company the size of CSL, this is in the context of a portfolio of projects across all stages of the R&D portfolio. So, you have your small biotech companies which are focused on one or two compounds, and that's their reason for being, just to move that compound through. You have the really large pharma and biotech which have, a massive number of projects in preclinical and the clinical, and then you have mid-size companies like CSL, which will have a portfolio that looks something like this. So this is taking our existing projects, splitting them up into those three categories that I talked to before, and into the phases from research here and through into phase 1 and then beyond. So I'm not going to describe any of these projects in particular detail, just to highlight the three that I am going to talk about. So there's the Factor 12A project. It's new product development. It's in the preclinical phase. CSL 362, also new product development in phase 1, and CSL 654, which is in phase 3 at the moment. And as I said, each of these three projects will give you a different insight into that process of moving from the bench to the bedside. Now, in terms of how we make our decisions within CSL to introduce new projects into the portfolio and then to progress them through from that introduction through the clinical phases and hopefully into launch, is that we have a stage gate process. And you'll find every pharmaceutical company will have something very similar. And this is about making sure that as you're moving your compound through development, you're stopping to sit back and ask questions. Are we making the right decision? Is this where we should be investing the money that we need to invest? I'm sure you've all heard about the amount that this can cost. You'll hear figures of $500 million to $1 billion to move a molecule from the start of this process through to the end. So given that cost, given that it's a competition for that funding within the portfolio, you've got to have some clear decision-making points. And so this is our stage gate process. Most of what I'm involved in and the research team is involved in is pre-stage gate 2. So looking at new projects and doing the research to justify going through this stage gate 2 into product development and then phase 1. But for the broader process, I guess this just looks like one piece in the puzzle. But if you break this out, it's actually a lot of different things that are involved in taking that initial molecule and moving it through to our stage gate 2 and then into the clinic. And I'm sure some of these terms are familiar to you, but we can break research out into target discovery, target selection, target validation. Having selected the target, it's about then generating a lead compound. From our perspective, that lead compound is a protein. From other companies, it'll be a small molecule. Having selected the compound, it's about the optimization of that compound so it's most effective in the clinic. And then it's about generating support for your concept, either in animal models and having moved beyond animal models, I guess doing what's now referred to as translational science, trying to understand the extent to which your compound is likely to work once you get into human clinical studies. And this is becoming a really important part of what we do, this translational science piece. And it's all about decreasing the risk prior to moving into those really expensive clinical phases. And I'll show you what I think is some important pieces of translational data that we develop. But just in terms of moving through that stage gate 2, taking your compound from preclinic through into the clinic, what are the sorts of things that we're thinking about? What do we have to have lined up in addition to a great molecule? So it's one thing to have a great compound, which is an antagonist of your target and has great effects in preclinical models, but you've also got to be thinking ahead. So when we get to that stage gate 2, it's about planning and developing strategies for how you're going to make your product, how you're going to manufacture it, the type of toxicology study you're going to do, thinking about the regulatory affairs process, and thinking about what the clinical development plan might look like. So it's not a detailed clinical plan, but you certainly have to have a concept about how you're going to move your preclinical compound through phase 1, phase 2 and phase 3 and then onto the market. And in addition to that, that sort of planning and strategy development, there's lots of other stuff you've got to do as well. So there's lots of legal, intellectual property, commercial assessments. They're generally preliminary views, but they're really critical to moving the compound through. So that's all the sorts of things that we need to think about as your compound moves from preclinical into the clinic, and that looks like a lot, and I guess it's reasonable to ask, well, just how long can that take? And the reality is it can take a very long time, and I think Ian Wicks touched on this in his seminar just a few weeks ago, but this is the timeline for the development of mavrilimumab, which is an antibody targeting the GM-CSF receptor. And as I think, as Ian mentioned, this receptor was first discovered, cloned by Nick Nicola and his group back in 1989. So that is a long time ago, 1989. And it's just now, in 2012, moving into a phase 2B. So that's more than 24 years. It doesn't have to take that long. I guess really the key here was the work done by John Hamilton, amongst others, between 1997 and 2002, actually developing the scientific data that suggested that targeting GM-CSF would be a good thing to do in a range of autoimmune diseases. Having developed that preclinical data, we, together with our colleagues at what was then Cambridge Antibody Technology, developed a lead molecule. There was a lot of work done in animal models and translational science between 2004 and 2008, phase 1 in 2007 and phase 2B in 2012. And hopefully that molecule will be in phase 3 next year and onto the market as a new compound for the treatment of rheumatoid arthritis, maybe in 2019, 2020. So from 1989, when Nick discovered the molecule, through to 2020 to get a new compound out there. So that's 30 years by my count, so that's quite a while. Look, I think this is something also that is important just to note. There's a lot of things that have to happen for your project, your compound, to move through from the preclinical phase into the clinic and then registration, and it's mainly about all of the different regulatory authorities you have to deal with. And I've given the example of dealing with the FDA here, but essentially you could do a very similar diagram for the European Medical Authority or the Japanese authority or any of the big authorities. So you can see here that to go from research through to an approved product requires multiple interactions with regulatory authorities, and you can fall over at any point in this process if you're not doing it properly. So it starts with the filing of an IND. Before you file that IND, you'll have a pre-IND meeting with the FDA where you'll talk about the science, how you're going to make the product, what your toxicology design might look like. Then you'll file the IND. That'll include, again, the science and the preclinical, the tox. It'll have now the phase 1 design. You'll have an iterative process with the FDA answering their questions, and then you'll move into phase 1. And then you'll, under that IND, move through phase 1, phase 2, phase 3, all of the time with each set of results going back to the FDA and asking them, is it okay for us to move forward? Do you think it's safe? Is this what we should be doing? You get to the end of that point and you'll want to file a BLA or a biologicals license application. This is really the critical phase. You'll have a pre-BLA meeting with the FDA. You'll make your submission. There'll be a series of questions and answers. The FDA might convene an advisory committee that will vote on your molecule. That vote's non-binding. The FDA can make their own decision if they want. And finally, they'll approve your product. And they may approve it, but you'll still have a lot of R&D to do because they'll insist on various post-approval commitments, further safety investigation and further clinical analysis. So it's a really big piece of the puzzle, one which I don't like spending too much time thinking about, but one which we have a very big group in CSL that's there to deal with this. So that gives you some idea of, I guess, the CSL context, the perspective that we have on development, and what it takes to move a project through those different phases right up to the interactions with the regulatory authorities. So I guess what I'd like to do now is talk about 3 projects in particular that will exemplify the pieces to the puzzle that we just talked about. And the first one of these projects is CSL 654 for the treatment of haemophilia B. So this is a pathway which I suspect that not too many of you have spent a lot of time thinking about since maybe first or second year biochemistry, but it's one that we spend a lot of time thinking about. And certainly if you have an inherited deficiency in one of these coagulation factors, you'll spend a lot of time thinking about it because it can cause some really serious diseases if you have that deficiency. The one I mentioned that we're going to talk about today is CSL 654, which is a recombinant coagulation factor 9. And if you have a genetic deficiency in that coagulation factor, you have haemophilia B, and you can see the consequences of that here. So this is a joint bleed in a patient that's been untreated with a coagulation factor. So this person has a deficiency in factor 9 and they haven't been treated and they develop these joint bleeds, and if they go through a lifetime of being untreated or either poorly treated, then the outcomes in terms of joint disease and a whole series of other consequences are really quite severe. Now, the way haemophilia B patients are currently treated is in a way that's described on demand. So when they have a bleed or they can feel a bleed starting or coming on, then they're treated with factor 9, and that factor 9 is either plasma derived or it's recombinant coagulation factor 9. The reason they're treated on demand is that the factor 9 has a relatively short half-life. Its half-life is around 18 hours, and that prevents you from treating them in a prophylactic context. You can't really dose up on factor 9, it becomes prothrombotic, so by the time you get the dosing about right, the half-life limits you to on-demand treatment. You can't prophylax with these types of molecules. So for us, when we looked at this, we've been working in this space for a long time, the unmet need was really to get better clinical outcomes for our patients and really to improve the quality of life by enabling prophylaxis. And so we thought, we and a number of other companies thought about the best way to achieve prophylaxis in haemophilia B patients. And the approach we took was to take factor 9 and fuse it to albumin. Now, I'm sure most of you will know that immunoglobulins and albumin both have a very long half-life in the order of, say, 15 to 30 days, and that's based on their ability to cycle through the FcRn salvage pathway. They'll bind to FcRn at pH 6 in the endosome, and then they'll be recycled to the cell surface, and that gives them this really long half-life. So the question we asked was, what would happen if we took factor 9 and fused it to albumin? Would we confer on the factor 9 the half-life of albumin? And you can see the molecule we designed there. So it's factor 9 with what we've described as a cleavable linker fused to albumin. So when the factor 9 is activated, and it's activated by a protease, either autoactivation or by other serine proteases within that cascade, you can see that the activation domain is cleaved out, and in that same process, we're cleaving off the albumin. This is at the site of clot formation, so it's a benefit to the patient. So this is the molecule that we generated, and it's expressed and made in CHO cells, and the first thing that we wanted to test was whether it will bind to FcRn in the same way that serum albumin or immunoglobulins do. And you can see here some biocore analysis where CSL 654 is binding to that FcRn receptor at around 1.6 micromolar. So that's telling us now that we have a factor 9 molecule bound to albumin, which is able to cycle through that salvage pathway. And in addition to that sort of work, we also verified that the factor 9 activity was intact. So moving through the molecule generation and optimization and into the animal models of disease, here's some data that was generated in haemophilia B dogs. So there's dog colonies with deficiency in factor 9, and you can test recombinant coagulation factors in these animals. And you can see here, looking firstly at the PK and then the pharmacodynamics, you can see the fusion protein here has a lot longer half-life than the wild-type molecule. And this APTT assay, this is a pharmacodynamic assay, essentially just a measure of the ability of the recombinant factor 9 to mediate clot formation in plasma sampled from these animals. The normal time to form clots in these assays is around 60 seconds. given the short half-life of the wild-type molecule, it's only a couple of days before you're getting an extension of that APPT time. And here with the fusion protein, you're really maintaining effective coagulation for a lot longer. So the curve runs below the 60-second mark for up to 6 days. So here in this animal model of haemophilia B, the half-life was extended by about 2.3-fold. The clearance was 4-fold lower and the area under the curve was 4-fold higher. And we observed similar effects in rats and in non-human primates as well. So that's really good preclinical data supporting the transition of this molecule through into the clinic. So a phase 1/2 study was designed where we did a dose escalation and PK study in patients with severe haemophilia B. The objectives were to assess those doses and really look at safety outcomes PK and safety outcomes and not so much efficacy in these patients. And just moving through quickly, I think the really pleasing thing to see is that half-life extension that was observed in the animal species really translated into patients. And so here you see in this group of adults with haemophilia B that for this molecule compared to, in this case, plasma-derived factor IX, you've got a massive increase in the half-life. You've got a 5.3-fold increase in the half-life and a 7.3-fold increase in the area under the curve. So the actual data looks better in patients than it ever did look in the animal model. So that was great phase 1, phase 2 data, and that prompted us to move this molecule into a phase 3 study. This is just a synopsis of what the phase 3 study looks like. There's actually 2 concurrent phase 3s, looking firstly in adults and then having demonstrated safety in adults, then in children. The study in adults is finished and the study in children is in progress. But you can see there's a day 14, 1 to 14 run-in where we're looking at the PK. Then from day 14 out for 6 months, we're looking at weekly prophylaxis compared to on-demand use. And in the next 6 months of the study, we're looking at extending that prophylaxis from 7 days out to 2 weeks. And then after 12 months, that's the end of the study. But in the extension period, beyond the end of the phase 3, we're actually able to look at extending that dosing interval out to 21 days. So patients are being dosed every 21 days. And I'm not going to show you the data because the data essentially looked the same as the phase 1, phase 2 data, where in the phase 3, and in both adults and children, we've really been able to demonstrate that we get a vastly increased half-life. And the PK profile indeed indicates that the factor IX fusion protein will be useful for prophylaxis treatment for intervals up to 14 days or longer. Now, this might not sound that important to you, and the technology is fairly run-of-the-mill, but I can tell you this is really life-changing for these patients. These patients who would have bleeds and would treat on demand are now able to have one intravenous infusion every 3 weeks, and that will prevent them from bleeding. So this is truly life-changing and a great step forward in the treatment of these patients. Okay. So that's I guess, a project where we're not talking about a new mechanism of action. This factor 12 was working in the same way as the other factor 12s. We'd modified the molecule to provide benefits to the patient, which they were really looking for. The next couple of projects I want to talk to are really looking at new mechanisms of action to treat diseases that are either reasonably well treated at the moment or not treated at all. And so the first one of these is CSL 312. So this is an antibody that targets factor 12 in the coagulation cascade, and it's been used to treat these 2 disorders here, and I'll speak a little bit more about these just now. So HAE is hereditary angioedema, and ECMO is extracorporeal membrane oxygenation. So just to recap, this is, when I talked briefly about the coagulation cascade previously, and there's two parts. There's the extrinsic pathway and the intrinsic pathway, and this is the intrinsic pathway or contact activation pathway, where you get damaged or negatively charged surfaces activating factor 12 to 12A. 12A then activates factor 11 to 11A, which activates factor 9 and initiates the whole cascade down to the formation of a fibrin-based clot. So that's the normal process, and the question we're asking is what happens when that goes wrong. So we're not talking about the deficiency of something in this case, we're talking about what happens when something's not quite right. This is one example of what can go wrong. So people who have either respiratory failure or cardiac failure will often be put onto heart-lung bypass machines for extracorporeal membrane oxygenation. So this is just getting the machine to do their breathing for them. And you can see one of the problems with putting people on these sort of systems, and it's often very young infants, is that there's lots of negatively charged surfaces and their blood is very prone to coagulating and blocking up the system. And the way that's dealt with is to give the patients heparin, and that stops the coagulation, but it puts them at great risk of bleeding and in fact bleeding out. And a very large percentage of the young kids and patients that go on to this sort of ECMO will succumb because of bleeding-related disorders. So this is really an important unmet medical need. How can we stop the coagulation without putting or giving the patients a bleeding risk? And as I'll show you, the way we've chosen to address this is by developing inhibitors of factor 12 and using those in this setting rather than heparin. The other piece of this cascade, which I didn't show previously, is that in addition to participating in the coagulation cascade, factor 12 is also a key starting point for the generation of bradykinin, which controls vasodilation and swelling or edema. And the way that works is activated factor 12, will convert pre-kallikrein to kallikrein, and the kallikrein will act on high molecular weight kinogen to release bradykinin, which acts on the bradykinin 2 receptor to induce vasodilation. And like all important biological processes, there's a lot of internal control mechanisms built in here, and you have things like C1 esterase inhibitor acting to inhibit factor 12 and to inhibit kallikrein. So that's a feedback process which will inhibit both of those mediators of this process. So what happens when this goes wrong? People are born with a deficiency in C1 inhibitor. And if you have a deficiency in C1 inhibitor, you are prone to having attacks of what's known as hereditary angioedema. And here you can see a patient having one of these attacks here, where they can have really dramatic swelling, in this case in the lips and the face, but these attacks can also affect vital organs and people can succumb and die. Particularly, for example, if the attack's focused on the larynx, you can suffocate. So here, in this particular setting, factor 12 is a key upstream mediator of hereditary angioedema. So the way these patients are treated at the moment is to use plasma-derived C1 inhibitor. But plasma-derived C1 inhibitor, like factor 9, has a very short half-life. So all of these patients actually are treated for an attack on demand. And I can tell you some of these guys will be having weekly attacks. So they'll have 50 to 60 to 70 attacks per year. That's not all of them, but it's certainly some of them. So they're treated on demand with C1 inhibitor. So is there a better way of treating these patients? So what I'm going to talk about next is the development of a factor 12 inhibitor to use both in this setting and also in the treatment of these patients. We did a lot of work to develop some human monoclonal antibodies that target factor 12 and inhibits this activity. We have a number of antibody generation technologies that are available to us, and for this particular example, we chose to screen a phage display library of human antibodies. And you can see here some of that data. This is probably summarising 1 to 2 years' work in one slide. But this is a small peptide called infestin, which is a known inhibitor of factor 12. And here's a series of antibodies that we're able to generate from the library. And you can see one of these antibodies, 3F7, was a very potent inhibitor of factor 12 activity. And you can see here, this is just looking across a bunch of species, and this 3F7 antibody worked well in mice, in rabbits, and in humans. It didn't work in rats. So a lot of work led us to the generation of 3F7, which showed complete inhibition of factor 12. Having isolated that lead candidate, we then engaged in a process of lead optimisation and in the case of antibodies, that generally means you're looking to further affinity mature the antibody so it binds to the target with improved affinity, or add in other characteristics that will give you benefits in terms of the disease you're looking to treat. So in the labs down at Bio 21, 3F7 was affinity matured. We came up with an antibody with a 60-fold improvement in affinity and a 5-fold improved potency. I mean, I guess the interesting thing here, every time you affinity mature an antibody towards a particular protein target, you tend to mature it away from other things that it's cross-reacting with. So because we were selecting for increased affinity against human factor 12, we actually lost binding to the mouse and the rabbit factor 12. So what all that means is that when we want to go back into animal models to do experiments, we use 3F7 as a surrogate antibody, but when we're talking about the human clinical development, it's the optimised CSL 312 antibody. So firstly, looking at models of ECMO. So this is just a schematic of an ECMO setting. I guess the important pieces here is that you have the pump here and you have the oxygenator. On either side of the oxygenator, you can measure a pressure gradient. So if the oxygenator, for example, starts to block from coagulation, you'll get an increased pressure on this side of the oxygenator. So this is what it looks like in humans. This is where we add 3F7 in at 7 mg/ml or heparin or saline. And obviously we can't do it in humans. We do it in rabbits. And when I say we, I mean our collaborators at the Karolinska Institute in Stockholm. And this is just confirming that 3F7 is in fact a potent inhibitor of rabbit factor 12. So this is the model, and we're looking to see whether the antibody will be effective at preventing coagulation. So you can see here, this is looking at the pressure on the appropriate side of the oxygenator. If you just use saline without heparin or 3F7, within a very short time frame, the oxygenator will block up and you'll get a big increase in pressure. You can see heparin down here in blue, you don't get any coagulation, and you can see with the 3F7 antibody there's no coagulation at all. And here we're actually visualising it, so this is electron micrographs of the oxygenator. You can see this is fibrin deposition on the oxygenator, so this is the initiation of clot formation. You can see with heparin, you know, a little bit of fibrin deposition, but really no clot formation, and 3F7 even looks better. And this is at 5 hours compared to 1.5 minutes. So 3F7 doing a great job at stopping coagulation, but is it putting the rabbits at risk of bleeding? And the answer to that is shown here. So when you've got coagulation in the oxygenator, obviously if you come along and nick the rabbit's ear, that you don't get any bleeding. This is what happens when you're treating those rabbits with heparin. The oxygenator is not coagulating, but look at the bleeding risk they have. So if you nick their ear, they will continue to bleed. If you treat with 3F7 specifically, there's no bleeding risk. So that's exactly the outcome we were looking for. We've got really effective anticoagulation in this setting without putting the animals at a bleeding risk. So a really nice outcome. This is looking at HAE. This is an animal model of HAE where, you know, it's a passive cutaneous anaphylaxis response. And I guess what we're looking here, we're injecting the animals, the mice with Evans blue, and we're looking for the release of bradykinin and vasodilation to cause the mouse ears to go blue. And in these experiments, you can see that having initiated that response, if we're treating with 3F7 and measuring the release of the dye into the ear, we get a very nice dose-dependent inhibition of dye accumulation. And in these experiments, we're really comparing it to some of the small molecules that are available to do this. So a really nice initial model to look at the 3F7 preventing the production of bradykinin. Here we're looking in animals that have a knockout of the C1 inhibitor gene. So these animals are really prone to vascular permeability, vasodilation. That can be actually enhanced using an angiotensin converting enzyme inhibitor. It's an angiotensin converting enzyme that breaks down bradykinin. So if you add that into the system, you increase the half-life of bradykinin and you get increased vasodilation. And to cut a long story short, you can see here that in these animals, there's a basal level of, this is looking at the colon, I should say. So you get a basal level of Evans blue leakage out into the colon. If you add in 3F7, you can completely block the release of dye into the colon. And if you use a control antibody, BM4, then you don't have any effect. So again, really nice preclinical data confirming that 3F7 is likely to be effective in patients that have HAE. And the next step here is that step of developing data from patients, and here we're looking at patient plasma and asking, can 3F7 inhibit factor 12 activation or inhibit the activity of activated factor 12 in plasma from healthy donors versus plasma from patients with HAE type 1. And clearly you can see that it has a profound effect on the activity of factor 12. So the current status of this program, having developed the candidate, having optimised the candidate, having developed the preclinical data and the translational data, is that it's now moving through into the clinic, into the toxicology phase and the manufacturing phase, and will be into the clinic shortly. So the final project that I wanted to touch on was CSL 362, and I guess I mentioned earlier that this is interesting because it's an example of a molecule that when it first went into the clinic, it didn't work. It didn't have an effect on the disease in the way that we'd hoped. So rather than abandoning the project, we decided that we would re-engineer the antibody and go back into the clinic. And I mean, that's often difficult to do. You do need the resources available to do that, but nevertheless, that's what we chose to do here. So what is this project about? CSL 362 is an antibody that targets the IL-3 receptor alpha, or CD123 as it's known, and can do two things, and I'll show you that in a minute. It can block the activity of IL-3, but it can also induce antibody-dependent cytotoxicity. And so the question is, why might you want to do that? So here we're looking at the distribution of the IL-3 receptor, or CD123, on normal cell populations, but also on a whole lot of AML populations. So acute myeloid leukemia, peripheral blood blasts, peripheral blood stem cells, leukemic stem cells, bone marrow blasts, and bone marrow leukemic stem cells. And you can see CD123 is really upregulated on these cell populations. And there's a lot of literature showing that these cell populations will survive and proliferate in response to IL-3. So it's upregulated, the target is upregulated on these AML cells, and these AML cells will respond to IL-3. The other important thing to note is, so this level of expression is higher than all of these normal cell populations, except for basophils and plasmacytoid DCs, which probably have a 5 to 10-fold increase in the level of CD123 expression. And that's good for two reasons. Firstly, we can use it as a biomarker population in people with AML. So when we're looking to treat patients with AML, we would expect also to kill and deplete their basophils and PDCs, and that allows us to track our mechanism of action in those patients. But the other, I guess, important thing, and the one that we're really interested in, is if you read the literature and understand how lupus develops, so this is systemic lupus, if you understand how that disease develops, it's pretty clear that particularly plasmacytoid DCs, but also basophils, are key cell populations in underlying the pathology of that disease. So it's thought to be an interferon-mediated disease, and the source of that interferon is plasmacytoid DCs. So if we can target and kill plasmacytoid DCs, maybe we've got a way of treating lupus in addition to a way of treating AML. And that's another thing we're always doing in pharma companies, particularly in the early stage research, is when we have a molecule, we're always asking ourselves, well, we want to treat this disease with it, but what else can we use it for? So here we started thinking about treating AML, but we've also got a whole lot of data that says we'll be able to effectively treat lupus. What I'm going to do today is just focus on this AML piece. AML, myeloid leukemia, is a very nasty disease. It's the most common acute leukemia in adults. I guess the key thing to note is it's almost invariably fatal. So patients, when they're treated with chemotherapy, with induction and consolidation chemotherapy, will invariably go into remission, but they will invariably relapse out of remission, and when they relapse out of remission, they will succumb to the disease. And here you can see what has happened in a leukostasis, in a relapsed patient where they have so many AML blasts circulating in their blood, they get leukostasis, and this is a coronary artery completely blocked up with AML blasts. And this just shows you that really there's been no new treatments developed in this space for about the last 20 or 30 years. So it is really a serious disease with a large unmet medical need. So I mentioned that we'd been into the clinic with a molecule that failed, and that was called CSL 362, and that was a chimeric antibody, chimeric IgG1 targeting CD123, and it had potent antagonist activity against IL-3. And at this particular point in time, the hypothesis was if you could inhibit IL-3 activity, you would be able to impact the course of disease because these cells were dependent on IL-3 for survival and proliferation. So really, antagonism of IL-3 activity was what we were looking for. We were looking in, this is the normal treatment regime or course for AML patients. And in this phase 1 study we were looking to treat patients with relapsed or refractory disease at weekly dosing at the doses you can see here. The primary objective was safety and tolerability and PK, but secondary objectives in this clinical study were looking at anti-leukemic activity and biological effect on CD123 stem cells. So this is just really summarising the outcome of that clinical study. So a 2-year clinical study, many millions of dollars. The hypothesis didn't really stand up, but you can see here that this is patient blood where we're looking at screening and then after 16 days. So you can see that looking at this patient's blast cell population and looking at phosphostat 5, you know, those cells really did respond to IL-3. And in the screening phase, if we added CSL 360 into that, we blocked that response. And indeed, in patients that were on drug for 16 days, we'd completely inhibited the biological activity of IL-3 on their blast cell population. And just looking at a control response to GM-CSF, we had no effect. But despite inhibiting IL-3 signalling, we had no effect on the course of the disease. So the hypothesis that this population was dependent on IL-3 was wrong, and in hindsight there were other, this was a redundant function of IL-3 and there were other cytokines that could step in like GCSF, GM-CSF and take over the role of IL-3. But the question asked, we did observe in that study that we had very good targeting of AML cells, so we could show all of the AML cells were coated and had CSL 360 bound to them. It's just that nothing was happening. So the question we asked, well, what could we do to 360 to make it more potent in this context? And what we thought we would like to do is actually to get that antibody to better engage the innate immune system to recruit NK cells, to get the NK cells to kill the blast cells and the stem cells. And the way we did this was using some FC engineering, which increases the affinity of the FC portion of 362 for these FC receptors you can see here. And the one to really notice is the increase in affinity for CD16. So the affinity of 360 was in the micromolar range, and after we'd engineered the FC domain, the affinity went up to 18 nanomolar, an increase of 112-fold in the binding of that FC to the CD16 on NK cells. And that really increased the ability of this antibody to induce ADCC. So here you can see, looking at primary AML cells, here's CSL 360, not really inducing any lysis at all, and here's CSL 362, really now able to generate substantial lysis in these in vitro assays. And here you can see, as well as targeting blast cell populations, the 34 positive, 38 negative putative stem cell population is also targeted as well. So we've got a new molecule now. We've been into the clinic, we realised what our problem was, we've come back out, we've optimised the molecule, and it seems to have increased activity. So we did a whole lot of work in the lab looking at characterising the antibody further. I mentioned previously that basophils and PDCs also express very high levels of CD123, so would 362 kill basophils and PDCs? So these are simple in vitro killing assays, whole blood incubated with CSL 362 for 24 hours. And you can see, looking at basophils, they almost completely disappear within 24 hours, and at this PDC population they completely disappear as well, and that correlates with the activation of NK cells as seen with the downregulation of CD16. So the expected result that we saw there. We did, there's not many good in vivo models of AML, but this is one we chose to work with in collaboration with Richard Locke in Sydney, where we're looking at transplanting patient AML cells into NOD-SCID mice. And without going into too much detail again, you can see that the addition of 362 really did give you a benefit in these animals, as you can see here. We do because of the lack of cross-reactivity of things like anti-cytokine and cytokine receptor antibodies in different species, you invariably end up doing experiments in non-human primates, and this is some work we did in cynomolgus monkeys, where we did a single administration of different doses of CSL 362. You can see here from 0.01 mg per kg up to 10 mg per kg, and in those non-human and in those primates, we followed the basophil and the PDC population over a period of around a month. And you can see, as was the case in those in vitro experiments, when we then went into an in vivo setting in primates, we got a very rapid depletion of the basophil population, and you can see that here, and also a depletion of the PDCs from baseline. So in vivo, this antibody is doing what we want. In primates, it's killing the susceptible cell populations. That's a really good thing from the lupus perspective later on, but it's not really telling us too much about what's going to happen to AML cell populations. And again, here we're trying to think of, well, what sort of data can we generate in the lab that will offset the risk as we move into the clinic? And this is an experimental design that we came up with to try and achieve this. So here, when patients first present at the clinic and are diagnosed with AML, we take a sample of their circulating blasts and we freeze them down. Those patients then go through induction and consolidation chemotherapy. And when they're in remission, we come back and take another blood sample, which is now pretty much normal levels of NK effector cells. And we ask the question, can those remission NK cells target and kill the diagnosis blast cell populations? And will it happen with the same kinetics at which we deplete basophils, so we can use those as a biomarker? So we're setting up these assays with a 3-to-1 ratio of effectors to targets. It's a 24-hour assay, and the data looks fantastic. So you can see over this period of time, the leukemic blast cell population really dramatically decreases, as does the basophil population. So this type of experiment has been repeated now in samples from about 20 to 30 different patients. And sometimes it's better than this, sometimes it's not as good as this, but the translation is there. We're seeing the killing that we observed in vitro and in the animal models translating into actual patient samples. And this is really what we want to see happening in the patient population. So this is what the 362 phase 1 study looks like, and it's just going on at the moment. So as distinct from the 360 study, where we were looking at patients that had relapsed and had active disease, now we're looking at patients in remission, and I'll explain why that is in a minute. The primary objectives are similar: safety, tolerability, but also PK immunogenicity and biomarkers of efficacy, looking at the basophils and the PDCs, but also in some of the patients, minimal residual disease. This IND was submitted on the 23rd of March. The study is ongoing in 4 different sites: Johns Hopkins, Seattle, Cornell, and here in Melbourne at the Royal Melbourne Hospital. Now, I showed you that treatment diagram before. So this is in the patient group that we were trying to treat with 360. So these were patients that had relapsed disease, were on the way out, and we're trying to rescue them from that situation. For 362, we've been able to think about it differently, and we're using a different paradigm. So we're not trying to treat that patient population here. We're trying to treat patients that are in remission and prevent them from coming out of remission. So it's a different way of thinking about disease. We're taking patients that have been treated with chemotherapy, they're in remission. High-risk patients in this group will inevitably relapse within a 6-month to 12-month time frame. And what we want to do is have 362 on board, so if they do relapse, then their immune system is intact still. They can recruit NK cells, they can kill any emerging disease, they can kill the minimal residual disease, and really extend out that time of remission, if not prevent relapse at all. So it's a different way of thinking about the treatment of disease, but it's based on the experience we developed both preclinically and then in the first clinical study. And just to finish up, these studies like this, getting from the bench through into clinic takes an enormous amount of work. The CSL 654 Factor 9 program was essentially an internal CSL program with people here and in Marburg and our clinical collaborators. CSL 312 anti-Factor 12, the lead CSL people were Con Panousis here at Bite21 and Mark Nolte, and they've done an extraordinary job in generating the data supporting this program. The main collaborator group there has been Thomas René and his team up at the Karolinska Institute. in Sweden. For the CSL 362 program, again, it's been primarily driven within CSL by Sam Busfield and Mark DeWitt, who's the clinical lead, but it's a long-term collaboration with Angel Lopez and his group in Adelaide, and more recently with our clinical clinical collaborators, and in particular, Andrew Roberts here at RMH and WEHI, and I'm sure you all know Andrew. So with that, I'd like to thank you for your attention, and if there's any questions, I'm happy to answer them.
Lovely to hear. Eliza, I understand you looked at glass. Did you look at glass? Obviously, it might 362 be useful now.
Yeah, it might. Mast cells are harder to look at, as you know. They don't circulate around in quite the way that basophils do. So we have tried to look at those in tissues, and we have some evidence that they are depleted, but it's hard to look at. So possibly, but we're not sure just at the moment.
Just out of curiosity, the new oral anticoagulants that target thrombin and some other molecule one of them targets, on the nose clinically because you can't reverse the bleeding that's been caused. Is CSL thinking of a way around? I mean, that's a perfect market for you if you can find the antidote that can treat the bleeding. I would have thought, because it'll be a massive market. You're not in that space at all? Or,
Well, it's strange you should ask that. It's like you've been sitting in on our R&D executive meetings. But look, those factor 12 and factor 2 inhibitors, the regulatory authorities have said to those companies, you have to identify a mechanism of reversing this coagulation effect. So we sell a product called Beriplex, which is one of these complex mixtures of plasma proteins. So Beriplex, which is used and approved to treat warfarin or to reverse the effects of warfarin, contains factor 7, factor 9, factor 10 and thrombin. And so that is being tested for the reversal of factor 2 and factor 10 inhibitors. And so hopefully it'll work. But it will require actually a whole clinical program to get approval for that indication.
do you think it's worth it? Presumably it is.
Well, it is worth it. You know, those other companies, Bayer and I forget who, they've come to us and said, can we try using Beriplex as a reversal for those agents? So something needs to be done, and I think that's probably the best candidate that I've seen.
Does CSL do much work on developing systems to deliver antibodies to the interior of cells?
No. I mean, we've looked at ways of getting peptides and proteins and things into cells before, and I've never found any of the data that convincing. I'm sure, you know, others will find ways to do that, but we're essentially focused on targets that are accessed at the surface of the cell.
I was interested about the reason why you used remission cells using the CD16 targeting antibody.
Yep.
And wondered whether or not NK cells from people with active disease, did you just not see any effects? Or?
Well, so people with relapsed active disease, their blood cell compartment is really dysfunctional and disordered. So they have very, very few NK cells. And if you relied on those relapsed disease NK cells to mediate any sort of killing by what's really an overwhelming blast cell population, I doubt that it would be effective. So the idea is to go back into remission patients where the blood cell compartment is intact and fully functional, hopefully, and try and rely on those intact functional cells to kill the emerging disease rather than kill the relapsed disease.
I'm also interested in the new approach to take patients that have gone into remission. How soon after the chemo do you give CSL 362? And was that determined sort of experimentally in small animals, or? No,
that was, I mean, those, look, you know, I don't know if Andrew's here, but you know, one of my clinical colleagues would better answer that question. But I think, you know, pretty much they're in there, they will have their induction therapy, they will have a greater or lesser amount of consolidation therapy depending on the site, and as soon as they've finished that therapy, they'll go straight on to 362. So some of them had induction and very little consolidation and end up straight on 362, you know, and others more consolidation.
And I suppose you've kind of answered this already, but does it make any sense to actually combine the two at the point of treatment? I guess you're going to say no because you're going to hit down K cells, but was that considered?
Yeah, look, maybe it might make sense to combine them, and maybe you might look at that down the track somewhere, I suspect, but we haven't looked at it in that much detail at the moment.
With CSL 654, what's the long-term rate of inhibitor formation with long-term prophylaxis use?
Look, it's hard. So, inhibitor formation in haemophilia B targeting factor 9 is much less of a problem than haemophilia A and factor 8. But we haven't seen any sign or issue around inhibitor formation in any of the clinical work that we've done. But as you probably know, it's a long-term phenomenon that you have to monitor, and we have to actually do those long-term studies to. But I wouldn't expect any problems with 654.
Thank you very much, Andrew, for joining me.